Oh, I see Lei here. Hey, Lei.
Hey, Darren. I'm from Tsinghua University. I'm very glad to be here to join this great meeting, and I will introduce two our presenters from our lab. They are all both freshmen from the lab of Xuegong Zhang from Tsinghua University. It's Erpai Luo and Chuxi Xiao. Maybe you can introduce yourself, and then let's introduce Devin to present our video. And after the video, maybe we can talk about some details of our work.
Yeah, I'm here. Can you hear me?
Yeah.
Yes, sounds good.
Right. Okay, so hi, Devin. It's really my great honor to be invited here to introduce the work of our group to you guys, and I hope you will enjoy it. Yeah, that's it. Yeah, I am very pleasant to be here. Yeah, thank you.
Yeah, okay. Can you hear me? Hello, Devin. I'm Chuxi Xiao, and thank you for holding such an exciting event. And because our members cannot gather together today to give an introduction, so we pre-recorded the video. And maybe we could play the video first, and if there are any questions, we could discuss after the video.
Sounds like a plan. The video length is 46 minutes and 30 seconds, so we'll just tune back right after it wraps up. All right, thanks again. I'll get this going for us.
Hello, everyone. I'm Chuxi Xiao from Xuegong Lab. It's glad here to give an introduction about our work, HECA, Unified Informatics Framework of a SEO Atlas. The title of our today's talk is Computational SEO Atlas Assembly. First, please let me introduce who we are. Our tutor is Xuegong Zhang Professor from Tsinghua University, and there are totally 31 members in our group who are interested in the area of life sciences and informatics. Okay, now let's begin our today's talk. We all know that there are many cells in our body, such as brain cells, liver cells, blood cells, and so on. Because there are many organs in our body, so the type of cells is also diverse. And the Human Cell Atlas aims to build the reference map of human body. But what should the map be like? This is a question for us to explore. So in order to solve this question, we revised the concept of HGP, that is the reference map of genes. The purpose of HGP is to determine the nucleotide sequence composed of 3 billion base pairs contained in human chromosomes, so as to draw the human genome map, identify the genes and sequences contained in it, and achieve the ultimate goal of decoding human genomic information. In order to draw such a map, HGP organized all multiscale information of genes on a linear DNA, and the linear DNA can form complex structures to regulate gene expression. Okay, we can say that making a SEO atlas is maybe similar to HGP, so maybe we can make a comparison between them. First, as for the goal of HGP is to build an atlas of all human genes, and for SEO atlas, it aims at building atlas of all human cells. And the fundamental element of them is gene and cell for each other. And what it is for HGP, it is a string of DNAs, and for cell atlas, it's a pack of molecules. And as for the size, we can see that the size of cell atlas is much more larger than that of HGP. And the basic alphabet for HGP is ACGT, and the atlas dimension is 1. But how about cell atlas? What are these for cell atlas? These may be also the questions waiting for us to explore. To go further, we can have a look on the cell atlas of elegance. There is an ideal cell atlas of it, because there are only around 1,000 cells with deterministic location and function in it, which means that we can say exactly where this cell is and what it can do. Each cell in it has a unique name or index and a cell type annotation. But does this fit human? Is human cell atlas also unique and ideal? The answer may be not. This is because that human cells are much more complicated. First, let's look at the number. There are around 37 trillion cells without explicit annotations in our body. And all of these cells have multiscale features, and they are interacting with each other. Next, the multiple functions of them are shifting with time and location, just as we grow up or as time goes by. Last but not least, our body is well organized, but the cell inside it has undetermined locations, which makes it even more difficult for us to draw a map of them. So how can we deal with these problems? Or in other words, what should a human cell atlas be like? There are totally 4 aspects. As for the big number without explicit annotations, maybe we should build with understandable and reusable annotations. And for the interaction between cells, we should store and explore a huge amount of cross-linked data, and this data could store the interaction between cells. As for the multiple function shifting with time and location, maybe we should keep all features that might be important for function illustration. And as for the undetermined locations, we should map cell locations on demand. And these are what we think the cell atlas should do for human. Besides, there is another lesson we can learn from HGP, that is the shotgun method. This term shotgun method originated from genome sequencing, which means that the genome is first interrupted, sequenced in sections, and then combined by a computer. Specifically, there are multiple copies of unsequenced genome, just as the first picture shows. And then we shotgunned these unsequenced fragments. These fragments then unsequenced and aligned. Finally, we reassembled them into a sequenced genome. To summarize, the basic idea of shotgun method is integrating the scattered data to get the final complete result. So, can we assemble cell atlas in a shotgun manner? That is, we can collect many scattered data from our real bodies and then assemble them into cell atlases. Based on this idea, we can assemble the scattered data sources with unified informatics frameworks and form them into a unified storage for flexible exploration and usage. As for the word flexible, we can make an analogy. Just as the picture shown on the left, we know that movable types is a technology that you can choose the word you need to put them in a complex sentence. One of the main ideas of it is choosing what you need to use. And this is the same with cell-centric assembly. we can regard a cell data or feature as a movable type, and then we can choose what we need to build a final assembly. This is what's the meaning of cell-centric and flexible atlas assembly, and this is also what we want to do. Okay, now we go to the next part. We know what a flexible cell-centric atlas is, and want to establish a cell atlas. But the number of these is truly big. You may not have that intuitive knowledge about it, but I can show you some comparisons to let you have a better understanding. You can see that for the genome, the magnitude of base pairs is around the 9th power of 10, with around hundreds of features. As for cell atlas, there are about 13 power of 10 cells in an atlas, each running a genome program. So making a cell atlas is much more difficult than making a gene book, which needs to store more data, and this may be the main difficulty for us to build a cell atlas. At present, most methods to deal with this are file-level collections, which means storing and indexing cells as a collection of files, just as the picture shown on the right. However, this is not what we want to do. To build a cell-centric atlas assembly with such a big feature, we need a really big width and big depth data repository for quick retrieval. Although now we already have some databases, they are not suitable for storing this data with so many columns because they cannot support such a big feature, and most of them are wide row based. But what we need is a wide column based. These databases can only store up to around 5,000 columns, but there are more than 40,000 genes and much more feature. To solve this problem, we proposed a novel unified giant table called UGT. UGT is a unified giant table for big data storage. It is a wide column database system for all molecular features and mental data of cells. Now we have the first version, which could store more than 1 million cell features and more than 100 million cells. And you can query cells quickly in UGT within just 10 seconds. If you still don't have an intuitive understanding about the function of UGT, I can show you a picture below to let you have a better understanding. You can see that in every row of UGT, there are key, some quantitative molecular features, and 16 mental data. These mental data include you have annotation, organ, study ID, and so on. By doing this, we can change the scattered data files into a unified giant table. Okay, now you may have known what UGT is and what it can do. But you may be wondering why it can outperform other methods. So now, I will make a brief comparison between UGT and other databases. Traditional databases are often row-based, so they have a small column limit. But UGT is column-based, so it has almost no column limit, though it may be more expensive to insert and update data. For this reason, traditional databases are more suitable for file-level collection, but not for cell-level assembly. Instead, UGT is already for assembly of more than million cells within more than 1 GB features on a desktop server, searchable in sub-seconds, and support multiple programming languages. And these are the highlights of UGT. So after building this framework of UGT, we now have the ability to store a big number of cells and the mental data of them. But another question comes: how can we annotate a specific cell from this big data? This is similar to annotating a specific person. There are many aspects that you can use to describe a person, and all of them are correct, but it can be confused. For example, just as the picture shown left, a person could be called an engineer, a building engineer, a senior engineer, or registered construction engineer. You cannot say any of these identities is false, but it can be truly confused if you are not familiar with these names. And this is the same with annotating a cell among a lot of them. There are some trouble as well. Here, I will show you some complexities caused by the source of datasets. First, the naming of single cells from different datasets is confused, and the cell framework for single-cell data is missing. Next, in an organ or tissue, there is a hierarchical relationship between cell types. The hierarchical relationship of these cells may lead to confusion in cell type labeling. Different single-cell datasets may label cells at different levels. For example, the sum cell is labeled T cell in some datasets, and the CD4 T cell in another dataset. Due to the existence of data hierarchical relationship, these annotations are all correct, but they will lead to users' misunderstanding. Last, when cells are labeled as different levels of cell types, it is obviously impossible to use the marker labeled by the same set of cells. Therefore, the single-cell marker genes collected from different data cannot be migrated into other datasets, and a marker system based on cell-level labeling is required. So, in order to solve these problems, we introduced a novel framework called UHAF, furtherly known as a Unified Hierarchical Annotation Framework, and the basic structure is as follows. UHAF has two main aspects, that is, microscopic entities and microscopic entities. The relationship among entities include is a, part of, and connected to. Also, UHAF is accompanied with corresponding annotation criteria. The basic graph of it is shown left. We can see that the macroscopic annotations is listed from organ system to suborgan, and the microscopic annotation is some detailed cell information and the features inside each cell. What's more, UHAF is complete and expandable. Complete means that it contains all existing annotations and the mental data information we collected now. And expandable means that it is easy to add future discoveries and future resolutions into UHAF. The main difference between UHAF and other existing methods is the difference between local marker and global marker. For instance, if we want to annotate a specific T cell and describe its characteristics. In global markers, it will describe its detailed features directly. However, in local marker, features are defined in each branch. That is, first define what the feature of this level is, and then define what the feature of next level is. This advantage is in line with our logic, because we can mark the detailed features step by step, instead of showing the final detailed features once. The reason for doing this is that we want to build a unified hierarchical annotation framework, and by defining the feature of cells hierarchically, it can help us to do better, cell atlas assemble easily, and be a framework that is more suitable for annotation. So to realize this hierarchical annotation framework, we establish UHAF database. Up to now, there are 135 cell types and 501 local marker genes in it. But this is not finished yet. In this database, markers are determined by aspects of each organ, and it will include more organs, more cell types, and more related features in the future. We also want to establish an API, which could help users to add the annotation information of cell type into other annotation tools more easily. This could enable users to use and convert information between multiple tools without obstacles. In addition, in order to ensure the extensibility of UHAF and make it compatible with rapidly changing cell types and marker tags, we provide an interactive page to revise UHAF. We hope that through the strength of the whole single-cell community, users can directly feed back errors and updates found to us when using UHAF. On the website, we provide operations such as moving, renaming, deleting, and modifying the local marker. After user submits the modification of opinions and provides the corresponding reference and evidence, we will receive and process these opinions, and finally extend them to the current UHAF through version update. What I have shown before are the functions and frameworks of HACA now, but that's far away from what we want to do. In the future, we want to include some other features. We all know that a cell has multiscale features, and now I want to first introduce what single-cell attack set is to you. When DNA in the cell needs to be transcribed, the high-level structure of DNA will be particularly unraveled, exposing the double strand of DNA, and the exposed chromatin is called open chromatin. Attack seg uses DNA transposase and high-throughput sequencing technology to study the accessibility of chromosomes, just as the picture shows, and it can infer gene regulatory network, compare different cell types or disease states, and help us to understand differential or developmental strategies. By adding attack seg into HACA, we can have a more comprehensive knowledge of transcription process in cells. But one of the problems is that the feature of attack seg is even much more larger than that of RNA. So this will lead to a more rigorous technical examination, and this is what we want to solve in the future. Maybe we could store not a count, but a peak region on a genome. Just as this picture shows, we can store some peaks, that is, where it starts and where it ends. Another aspect that we want to add into HECA is extracting spatial information of cells. The expression of some genes can reveal the spatial structure of the tissue and provide location of cells. These genes are called spatial variable genes, and we want to add the information of them. To do this, maybe we could develop an algorithm to map the position of each cell to each slice, so that we can extract its position information and restore it to the slice. And now, we have already developed a transfer learning-based tool named STEM to achieve this goal, just like the picture showing right. Okay, then let us welcome Erpai Luo in our group to introduce some other information about our work.
Hi everyone, I am Erpai from the SG Lab. Thanks Chuxi very much for the wonderful introduction of UGT and UHAF. So now let me present to you guys the main role of tonight, and that is the Human Assemble Cell Atlas. Based on the UGT and UHAF, we built this Human Assemble Cell Atlas and try to make a better use of the single cell data. So as you can see, the HECA is a platform for assembling single-cell data into a unified giant table. It's kind of like an exploration for building an effective information framework, and we hope that it can help us better utilize cell data and cell atlas. So here, let me introduce the detail of HECA to you. The first thing is that the HECA contains a lot of cell data. It assembles more than 1 million labeled human cells from more than 100 published datasets, covering 38 organs and 11 systems. And that is basically most public human cell datasets you can find in the web. Besides the huge amount of cell data, we also invited 2 new methods of Atlas application based on the cell-centric assembly, and that is portraitures and in-data cell sorting. Well, first is the portraitures. Okay, so what is the portraits of biological entities? Just like a real geographic position, which has multi-level information such as you can see on the left, like state, like city, and street. Our body can also be described in multi-aspect. So, just based on this idea, we propose the concepts of portraits of biological entities to describe the body from different angle, and we build this concept into our HECA. So you can find in the HECA that it contains a set of quantitative portraits of the biological entity, including, as you can see on the right, including organs, cells, and gene level, and for all quantifiable characteristics at multiple angles. So in the portraits, the first level is the organ level. You can consider it as a city in the body kingdom. So here, the anatomy relationship describes which state it belongs to, and also its relationship with other cities. And the position on UHAF show the detail of its position. And here the cell type is kind of like the industrial composition of the city. And each cell types corresponding to a specific industry, through which you can tell what kind of city it is, or what kind of cell it is. And the embedding viewer here can give you the overview of this specific organ. And next is the cell portraits. So remember that all cells from the same type is kind of like a factory in your body. And here, the origin organs show the distribution of this industry in the body kingdom. And also the marker gene tell what kinds of equipment does this factory has. And this is also an indicator which shows why this factory or why these cells has the ability to make certain products. And last, as for the gene portraits, we offer a detail about the specific equipment and also its working condition in different cities. And we believe that this is not enough. So in the future, we will add more description, like temporal information. As you can see on the right, like the developmental stage of the cells, or the states of the cells, to make the whole portrait more vivid. We hope that this will give us a deeper insight about our body and help us to uncover the mystery of some, uncurable disease. And that is all about the portraitures. So before we go into the in-data cell sorting, I would like to introduce to you first about the ECOT. And what is ECOT? As it is pronounced, is an application program used for catch certain things. And this is the tool that makes the in-data cell sorting come true. And as you can see in the picture, we are not here to catch the Mr. Bean. We use the ECOT to catch the specific cells that are meaningful for our research. And this is how our application program looks like. It, as you can see, looks very simple, and it's also very easy to use, both in the web and also through the code. Just click your mouse for several times, or 3 rows of code. You can catch all the cells you want in the database. Okay, now is the in-data cell sorting's turn. It's the core function of ECOT, and as you can see in the left, people usually sort out the desired cell with physical methods like lasers and electric methods. This can surely sorting the cell according to their physical properties. But here we sort out the cell from the UDTs in a much powerful and relaxed fashion. Imagine that you walk into a restaurant and at GCA tonight. You suddenly feel tired about this potato chips and spaghetti. You want something different. So you tell the waiter, "Hi there, I want some T cells in the heart with normalized expression of gene CD3E, alright, CD3E greater than, let me think, 0.5, please." Okay, if the waiter you are facing is an ordinary hothead, then his reaction will probably like, "What?" But remember, you are in the restaurant of HECA. The waiters here have ECOT. So with the help of ECOT, we can search from the unified giant table and offer you the meal or T cell as you wish to you. And this is the things in data cell sorting do to quickly sort out the desired cell data from the UDT database. So now we know what the ECOT can do and what the in-data cell sorting is. But what is the meaning of ECOTs and in-data cell sorting? A very intuitive change is that because of its ability to obtain the desired cell according to certain conditions, ECOTs can greatly reduce the complexity of biological cell research, and which means that enabling researchers to carry on virtual cell experience and easily test these hypotheses and assumptions so that they can focus more on the analysis of the phenomena rather than just working on this boring biological cell experience day by day. And another usage of ECOT is that it can help us to build an annotation reference. This reference is very much like a cell wiki, which can give you the information about some unknown cells. So how can we build this wiki? First, we import certain cells we like or we are interested in into it to build our special reference. Then the next time when you come across some unknown but similar cells, it can recognize them and annotate them automatically based on the reference you have built. So now we have talked a lot about what is ECOT and what the in-data cell sorting can do. But words alone are no proof. Let's have something real. So here we introduce to you about the application of in-data cell sorting and the mission of CAR-T off-target search. The CAR-T is a newly developed treatment for cancer. It's like a very powerful weapon that can eliminate all the cells with certain gene expression, including these cancer cells. But as a good soldier, we don't want to hurt these innocent cells. So we need to find out the gene that is expressed mainly on cancer cells. Here, with the help of ECOT, we can accelerate the process of evaluate off-target gene through virtual experiment. Let's say find out how many targets are innocent, or if the target is expressed but specially by these cancer cells. And here we use the in-data cell sorting to find an off-target in the brain for the brain suffer problem. You can see clearly, very clearly about how the gene expressed in different organs, and target gene is you can see this target gene is also expressed in the brain, which could make the healthy brain cells suffer. Alright, now we have comprehensively introduced the HECA and also its components to you. Next, let's take a further step to explore its ability. First, I would like to present the Unicode to you. It's a general machine learning framework for representing cell features. Okay, so we know that we live in a digitalized world now. Everything has become numbers now, like identity, like bank accounts, internet records. If privacy issues are not to be considered, it's no doubt that this digitalization did bring us a lot of convenience. And the same thing happened in the field of biological information. The digitalization could be a great benefit to help us dig out super valuable things from data or from numbers. And the Unicode here is a tool to dig out the attribution of the cells. And we use it to help us describe the cell's attribution in a structural and mathematical manner. And let's say we wanted to use it to digitalize the cells. We use a vector here to contain multi-phased feature of the cells. And each cell in a vector could stands for a certain attribution of the cell. In this way, we can represent and explore cell function within a mathematical space, and also maximize the capability of machine learning for future research. Because everyone knows that the machine learning is very good at with these numbers and data. And the next things here is the GRP Draw Pass. It's a tool that can help us to reveal the causal relation between gene mutation and disease phenotypes. As you may know, serious diseases are often caused by a tiny genetic variation, like a butterfly effect. So what we try to do here is to find out what's going on after the butterfly flipped its wings. What is the middle or inter steps looks like. Here, with the help of GR Pass, we can now use a variety of data to unfolding the black box of disease. And after we know how this illness come into being, we can find possible treatment to fight against them. Like this little experience we have took, you can see how a genetic mutation in some certain kinds of hot cells, affect the chromatin accessibility and change the state of cell, which turn out to be a serious heart disease. What's more, we believe that there could be much more possibility for our Edge ECA in the area of artificial intelligence, like the recent advance in BERT model. As you may know, BERT is a Google-created deep learning model to deal with neural language problem. It has now been widely applied in the field of like question answering system, a search engine, and our daily application. So one of the secrets behind this huge success is the massive data they use to train the model. And now, with the tremendous amount of easy-to-use cell data in the Edge ECA, we can modify it and train the BERT model to extract abstract information from cells. So, with its powerful representation ability, we can now examine the cell with an unprecedented clarity. Or in another way, we can now see through the cells. After we've done this, we can use the information to carry on many kinds of downstream work like cell classifications. Okay, and that is basically all about the Edge ECA. Let's make a quick summary. So we create a unified information framework for single-cell data, and we develop this framework into Edge ECA as an assembled cell atlas. And so far, we have created many powerful applications in the base or with the help of Edge ECA, like the in-data cell sorting and other tools like Unicode and GC Pass, Draw Pass. So we are looking forward to have more valuable tool or scientific discovery with the help of HECA. Right now, we have finished the introduction of HECA and its relative tools. Now, let's take a look at what we're going to do next. Our ultimate goal is to fully develop HECA and make it a complete and expandable cell atlas. We hope that this atlas will not just a map of our body, but instead, I would like to say it in a more science fiction way, a simulator of our body, from which you can tell the detail of your body consistence, including all the information about every type of cells, and what's more, all the interaction and the relationship between them. Just like the city in the game of SimCity. We wanted to make these cell cities alive. And what can we do after we finally got this map of human bodies? So if you have ever watched the movie The Matrix, remember that what Neo has saw after he became the One, all the people in digital form. And that is what this map useful, to help the scientists and doctors to become the One in the real world, to find out the secret behind our body and solve this incurable disease. To be more specific, as you can see on the right, we wanted to help us to like invent new drugs, to develop better treatment, to find out more efficient detection methods for disease, and other possible applications. So these are all the contents that our groups wanted to introduce. We wanted to thank Professor Katie Brunner for inviting us to join this fabulous great event. And our program is supported by National Key Program of China. And we're now having a bigger team across 10 institutions in China. We aim to constructing and using cell atlas in a better way. So you are very welcome to exchange ideas and maybe cooperate with us. So that's it. Thank you very much for listening.
Excellent video. I wanted to, did we get any questions in the meantime? I do have one. If not. Yeah, I wanted to ask about the use of GR path to identify these causal regions. Could you go into that just a little bit more?
The GR path actually try to link the mutation and the phenotype in a putative way. So we try to. Sorry, I will show the.
No worries. Take your time.
Okay. Yeah. So we try to integrate the data from GWAS and GTEx and some other single cell RNA-seq data in HEC to revolve the causal relationship between mutation and the phenotypes. Actually, we try to use some statistical methods to link the genetics variants to chromatin accessibility, because some non-coding variants usually influence chromatin accessibility to regulate the gene expression of other proteins, and then cause the some abnormal phenotypes. So we build statistical methods in each arrow. So from variance to chromatin accessibility, and from chromatin accessibility to gene expression. And these three sections are based on the GWAS results and the GTEx data. And then we use the single cell RNA-seq data from HEC to try to link the abnormal gene expression to some cell types. Actually, the GR pass will figure out many paths as the figure shows, but we will give scores to each path and tell you that which path will cause the phenotype most likely. And then here are some examples. Two types of heart disease. We find that mutation influence the expression in smooth muscle cell. This gene ARHG AP42, and then the smooth muscle abnormal smooth muscle cell will cause the heart failure. So this is the basic concept of this work. And I will mention one more thing, because here we only use single cell RNA-seq data, but now we are trying to integrate single cell RNA-seq data and single cell ATAC-seq data, as the ATAC-seq data actually mirror the chromatin accessibility. Now the interaction between variants and the chromatin accessibility is derived from prior knowledge, but now we want to build some more interesting model that use more big data to gain more accurate results. So it's a future plan of our HEC Cell Atlas application.
What are the next steps that you see for your team?
The next step, we will try to update the HEC, the Human Example Cell Atlas, that will involve multi-omics such as ATAC-seq data, as I mentioned before, and spatial omics. And we will integrate more machine learning tools to make the users to explore the function of gene cells and organs more efficiently, such as we will introduce the Unicode into the cell atlas to provide each cell coordinates, to make people don't need to run the machine learning method by themselves, but just query all the results from a website.
Thank you so much, Lei, for answering these questions, and thank you everyone on the team for putting together the slides and presentation. I'm excited to share this one. Thank you again.
Thank you, David.